Hello everyone. In today's video, we are diving into the world of bioreactors, specifically the continuous stir tank reactor, commonly abbreviated as CSTBR. It plays a crucial role in biochemical and fermentation industries. So, grab a note and let's begin. Introduction. A continuous stir tank bioreactor (CSTBR) is a widely used type of bioreactor in industrial microbiology and biochemical engineering. It operates under continuous conditions where fresh nutrient medium is continuously supplied and an equal amount of spent medium containing cells, metabolites, and products is continuously removed. This ensures the volume inside the bioreactor remains constant. CSPR are commonly used for microbial and enzymatic processes due to their efficient mixing and ability to maintain a uniform environment. Process. In a CSTBR, the working volume is kept constant. Nutrients are continuously introduced into the reactor and products along with microbial cells are continuously removed. The reactor content are uniformly mixed using mechanical stirrers to achieve homogeneity in terms of concentration, temperature, and other operating parameters. This setup enables a steady state condition to be achieved where the microbial growth rate matches the dilute rate. Diagram explanation. The diagram of a CSTBR typically includes the following components: feed line for introducing sterile nutrient medium, stirring mechanism to maintain homogeneity, sparger for an aeration in aerobic processes, thermostatic control for maintaining desired temperature, sampling port for withdrawing the cultural samples. Effluent line for continuous removal of biomass and product. The reactor is cylindrical, usually made of stainless steel and equipped with sensors to monitor pH, temperature, dissolved oxygen and agitation speed. Let us look at the assumptions. First, perfect mixing. The concentration of all components is uniform throughout the bioreactor. Second, constant volume. The rate of input equals the rate of output. Third, steady state operation. The system reaches and maintains a steady state. Fourth, no product inhibition or substrate inhibition unless specifically modeled. Fifth, biomass is uniformly distributed. Sixth, constant temperature and pH conditions. Model development. The model is developed based on mass balance principles. We consider the rate of accumulation of a component within the reactor as the difference between the rate at which it enters and the rate at which it leaves, plus any rate of generation or consumption within the reactor. According to the total continuity equation, this equation represents the overall mass balance in the reactor. Fi is the volumetric flow rate of the inflow, F0 is the volumetric flow rate of the outflow, Rho is the density which is assumed constant, V is the volume that is also assumed as constant. Since Rho is constant, this is the equation that we have simplified. Next, we will move on to component continuity equation. These equations focus on the mass balance of individual components, specifically biomass X and substrate S. Concentrations are typically used in these balances for well-mixed systems. For biomass, it is the accumulation where this represents the rate of change of the total amount of biomass in the reactor. Inflow, where Fxi is the concentration of biomass in the influent. Outflow, Fx, where X is the concentration of biomass in the effluent. Next, generation, where Vrx, where Rx is the rate of biomass growth per unit volume. Putting all together, we form this equation. Let us go to the substrate. Here, the accumulation where dVs by dt is equal to V ds by dt. This represents the rate of change of the total amount of substrate in the reactor. Inflow, Fsi, where Si is the concentration of the limiting substrate in the influent. Outflow, Fs, where S is the concentration of the limiting substrate in the effluent. When we talk about the consumption, is the rate of substrate consumption per unit volume, which is denoted by Vrs, and we take Rs. Putting it all together, we form this equation. Next, we talk about the reaction kinetics. This tells us the rate of the new plant growth depends on two specific things, which is mu, that is the specific growth rate, and also the x. Y is the yield where mass of B produced by mass of A consumed. Here, in simpler terms, the definition for reactor kinetics is, which is a part of our model and describes how fast the microorganisms grow and consume their food. The growth rate Rx depends on the specific growth rate mu, which is like how happy and fast each individual organism reproduces and the current number of organisms. Explain the monod model. The monod model describes how the specific growth rate mu depends on the substrate concentration. Mu is equal to mu underscore max into S divided by K underscore S plus S, where mu underscore max is the maximum specific growth rate, K underscore S is the half saturation constant. This equation is used to link growth rate with substrate availability in the CS2BR model. It is substituted into the mass balance equations to solve for substrate and biomass concentrations at steady state. Now let us look at the advantages. 1. Steady production for products over time. 2nd. Continuous operation reduced downtime. 3rd. High productivity and efficient use of resources. 4th. Better control over environmental conditions. 5th. Suitable for large-scale industrial processes. The disadvantages are risk of washout at high dilution rates, susceptible to contamination due to continuous operation. 3rd. Complex control and automation systems required. 4th. Less suitable for processes needing long adaptation or lag phases. Finally, we come to the applications. The application of CS2BR is industrial production of antibiotics, alcohol and enzymes, wastewater treatment through microbial digestion, continuous fermentation in food and industries, example beer, vinegar. Last, cultivation of microbial biomass for single cell protein. Thanks for watching. Hope you found this video useful.